Yes, ma'am.
All right. Okay, everyone, please switch on your video. Let's start today's class. Yes. So in the previous class, we were talking about the experimental verification of the chromosomal theory of inheritance. We talked about variety of things. Now tell me if anyone has any doubts from anywhere. Any topic you people got stuck while you were studying?
Yes,
Or
ma'am.
no? Clear? For everyone?
Yes.
Okay, very nice. You people study once the classes are done? Do you study? You do? Everyone does? Sure. Okay. You people have such trustworthy faces that if you say yes, I'll believe you. Okay. So now we will start with a new topic, that is linkage and recombination. Now see, these are the properties of chromosome. Now let us see how. See, the way the genes are the functional unit of DNA. The way the genes are present on the chromosome, like their physical association, their physical location and the presence of the genes on the chromosome is what we call as linkage. Okay, like they can be tightly linked on the chromosome, they can be loosely linked on the chromosome. Okay, and the recombination is when the non-parental character or the non-parental gene is like a new combination is made. It happens during the crossing over. Now, if you people again remember that cell cycle that I talked to you about in the previous class, right? There was this exchange of genetic material that takes place, and that is nothing but the recombination process. Where does it take place? So it takes place during crossing over. Now, the process of recombination and linkage, they go hand in hand. How? First of all, we'll see this thing that for like, at that point of time to study about the linkage and the recombination, T.H. Morgan, he was studying the Drosophila. The Drosophila had 4 pairs of chromosomes, out of which 3 pairs were autosomes, meaning their body chromosomes, and 1 pair was the allosome, that is the sex chromosome. So what Mendel did, that he carried out a dihybrid cross in the Drosophila. And with that, what did he do? He crossed the yellow-bodied and white-eyed female, meaning the Drosophila that had a yellow body and white eyes, right? The female Drosophila. He crossed that female Drosophila with the brown-bodied and red-eyed male. The ratio at the end of the dihybrid cross, it was very different from 9:3:3:1, meaning it did not follow the Mendelian ratio, the dihybrid cross ratio. So remember, why? Because, see, if the genes, they are very, very tightly linked on the chromosome, then they will show very low recombination, meaning they will not get exchanged very easily. Imagine they're so tightly fixed that they're not moving. But if the genes are loosely located, then it will show high recombination. So during that process, he saw that with the tightly linked gene, the recombination percentage only came out to be 1.3%, and that is it. But with the loosely linked gene, the recombination percentage came out to be 37.2. Now you see the difference by yourself, right? If the genes are very tightly associated on the chromosomes, they will not be very easily crossed over, or recombination frequency will be very, very less. Whereas the recombination frequency of the loosely linked genes will be much more. Now, Alfred Stuart Event, what he did that, okay, let us take the recombination frequency, like whatever is the percentage, to measure what is the distance between two genes, whether they are closely situated, whether they are not so closely situated, And with the help of this data, what he did, he started to map the genes, like the position of the genes on the chromosome. So like how the position of different continents, different countries are mapped on the globe or the map. Same way he wanted to map out where the genes are present. Now, when the mapping is done for prokaryotic organism, the unit is map unit. And when the mapping is done for the eukaryotic organism, the unit is centimorgan. Clear? Any doubts?
I might understand what mapping is.
Map, it's like a unit, like how we measure distances in terms of kilometers, right? Like how we measure shorter distances, like this thing, meters and centimeters. Same way, the distance or the unit for mapping of the genes on the chromosome is like for prokaryote, that unit is called as map unit, and for eukaryote, it is called as centimole. Clear? So when the crossover, like crossing over is happening, meaning the recombination is taking place. So like if 1% recombination happens, that is one map unit. If 2% happen, that is two maps unit. Clear? So that is how he found out the distance between the genes. Any doubts? Also, everyone, please switch on your videos. You're not supposed to keep it off.
Ma'am, can you repeat the last part again? Your voice lagged.
Oh, this part. See, whenever the crossing over is done, if let's just say the recombination frequency or the percentage came out to be 1%, then that is equivalent to one map unit. Imagine it like 1% is equal to 1 centimeter with that length, or 1 meter, that length, whatever. Okay, so same way in terms of the prokaryotes, it will be considered as one map unit. That will be the distance or the position of the gene on the chromosome. The same way, the percentage will increase and so will the map. Clear?
Yes, ma'am.
After that, there is the process of sex determination. Now, sex determination is the process wherein we can know the sex of the individual with the way the inheritance or the genes they are crossing. Right? So, establishment of the sex of an individual through different ways is called as sex determination. Now, it is very different from where, like for different organs. For example, in the case of turtles, crocodiles, and alligators, what happens is that in them, the sex is determined by incubation temperature, meaning however much time the incubation will be provided, the heat will be provided, depending upon that, the sex will be determined. Okay. Whereas in the case of Drosophila, in the case of birds, in the case of butterflies, honeybees, humans, grasshoppers, in all of these organisms, the sex will be determined by the genes. The genes will be responsible for determining of like the sex of these organisms. So there are two things. There is this phenomena called as heterogamety. Heterogamety is a phenomena wherein different kinds of gametes will be produced by a single individual. So it is a phenomena of production of different kinds of gametes by an individual. So one individual can produce two different types of gametes. So that case is what we call as heterogamety. So this heterogamety can be found. Yes, Aisha?
Ma'am, in the different types of gametes are the male and female gametes specifically.
Yes. Gametes are male and female gametes always. So, for example, if the male is making two different types of gametes, so that is male heterogamety. If the female is making two different types of gametes, then that is the female heterogamety. So heterogamety is a phenomena, normal. It can be present in the male and it can be present in the female also. So it is a phenomena wherein two different kinds of gametes are produced by a single individual. For example, okay, see, here are some discoveries. So, like, if you want, you can remember them. Henking in the year 1891, he discovered the X body, the X chromosome. Then Stevens in the year 1902, he discovered the Y chromosome. And Wilson and Stevens together, they said that the X and the Y chromosomes, they are the sex chromosomes. They are not the body chromosomes. Now, moving on to the heterogamety topic. So when we talk about the first case, that is male heterogamety. So in this case, the male will be producing two different types of gametes. Okay, and whichever, like for example, if it is male heterogamety or female heterogamety, so in either of the case, the male and the female will be responsible for the sex of the offspring. So when we talk about the male heterogamety, so it is the sperm, the male gamete, that will be responsible for the sex of the baby or the offspring. So under the male heterogamety, there are two mechanisms. First is XO type and then there is XY type. Now XO type is present in the grasshoppers. So here what happens is that the male produces XO and the female produces XX. So in either of the case, the male is going to, like for the gametes, it will give X or it will give O. But the female has no other option. She will be giving the X gamete. So when the X of the male and the X of the female combines, that is a female offspring. When the X of the male combines with another X of the female, that is again a female offspring. But when the O of the male combines with the X of the female, that is the XO situation and it is the male offspring. Clear?
Yes, ma'am.
Yes. And lastly, again, the XO situation is when the male will be there. So these are the cases of male heterogamety and this XO type is found in the grasshoppers. Okay? This one. Okay, so XO and XX type of situation is there in the case of the grasshopper. Now the second case is the XY type. Now in the XY type, this one is found in the Drosophila and mostly all the mammals, including the human beings. So here, the male is heterogametic, that is XY, and the female is homogametic, that is XX. So when the X of the male combines with the X of the female, the offspring is female. When the X of the male combines with the X of the female, again female. But when the Y of the male combines with the X chromosome of the female, then that results into a boy child. So in the case of humans, 23 pairs of chromosomes are present, out of which 22 pairs are the autosome. Autosomes means the body chromosomes, normal. One pair is the allosome, which is the sex chromosome. And this is the one pair that is responsible for the sex of the offspring. Clear, students? So male heterogamety is clear to everyone, both the examples.
Yes, ma'am.
Then comes the female heterogamety. Now here, the female will be responsible for the sex of the offspring and not the male. So the female will be producing two different types of gametes. Here the two mechanisms are ZW and ZO type. So ZW is the female and the ZZ is the male. And this one happens in the case of birds. Whereas in the case of butterflies, the female is ZO and the male is ZZ. In both of the case, the female is heterogametic. And remember, in whichever is the case, male is heterogametic or female is heterogametic, in either of the case, the one that is heterogametic, they will be contributing to the sex of the baby. It will be them or their chromosomes because of which the sex of the baby will be decided. Clear, students? Here, the most important thing to remember is the type and their example, like which organism has this type of sex determination. Now, talking about the most unique sex determination that takes place in the case of honeybees. So now, in the case of honeybees, the sex is actually decided depending upon the number of chromosomes the organism is receiving. For example, if it is a female, so firstly, we will learn about the different classes of honeybees. So there are three different classes of honeybees: queen, worker, and drones. Now, queens are the females, the workers are the females, and the drones are the male. So these are the classes of honeybees. Classes of honeybees means the different types that are found. Now, once the male gamete fuses with the egg, in the case of the honeybees, the offspring is naturally a female. Okay, now this female can become a queen or a worker depending upon the diet that they have. But genetically, they are purely female. But whenever the offspring is developing from unfertilized egg means the egg did not get fused by the sperm. The egg was just like that, and then it became the male individual. Then in that case, the male will be haploid. Here, the female will be diploid because it is formed as a result of fusion. But in the case of the unfertilized egg, the male will be haploid. And this process of development of an organism from unfertilized egg is what we call as parthenogenesis. Clear? Does anyone have any doubt? Now, in the case of honeybees, they have 32 chromosomes, right? The females will be diploid after the fusion of egg and spawn, and they will be having 32 chromosomes. But since the male is like the males are formed because of the unfertilized egg and not because of fertilization, so the male, they will be having half the number of chromosomes as compared to the females. So the males will be haploid. Clear, students? And this type of sex determination that takes place in the case of honeybees, we call it as haplodiploid sex determination. Haplodiploid sex determination.
The males have 16 chromosomes.
Yes.
The of unfertilized egg.
So like now the male has 16 chromosomes. So this male gametes, they are formed by meiosis or mitosis?
Mitosis.
Meiosis.
Mitosis.
You
You people are
obviously
right. You people are right. If I make faces, then that doesn't mean you people are wrong. I will make face. You have to be confident on your answer. Okay, you are very, very correct that it will be formed by mitosis. Now, please explain it very nicely.
Meiosis happens for like male and female gametes.
Okay.
When there are male and female and it does fusion, and then we get an offspring that is diploid. But here it's
Normal condition. Here,
here the
complete.
offspring is haploid. The male egg is haploid, so that's why its process is done by mitosis and not meiosis.
But why not meiosis? No. Listen, why not meiosis? Now, you people said mitosis, you might have thought about something, then only said, na, or guessed. You might have thought about something. No, didn't think. Why? Okay, why the gametes are formed because of meiosis?
Because there's fusion.
No.
Oh.
Before that, go one step behind. Fusion is later on.
Because the
Hmm,
the
hmm.
gametes should be haploid, and
Yes.
then fusion happens, and then
Hmm,
the zygote will be usually like diploid.
hmm.
But then
So here,
the haploid gamete
hmm.
becomes male. And
So, but like, okay, you are right up until now that meiosis happens in a diploid cell because we have to, like, the gametes have to be half their chromosomes, right? They have to be haploid. So why is meiosis not taking place when the male drone has to make its gamete? It's still the gamete formation. So why not meiosis? Just think a little bit more, this much more, and your answer is there.
There is male and female for the
It
meiosis.
doesn't matter. I am not talking about fusion. I am for fusion, male and female are required. I am talking about just the gamete formation. Why? You people see, you gave me like the tough thing that I asked, you gave the answer. Now the easy thing that I'm asking, you're getting stuck there.
It happens for early like somatic cells, reproductive cells. The
So
meiosis.
this is somatic cell now only. Now they are like now their like meiosis will happen to form the gametes later on, right? So the only question comes along here that females, they will undergo meiosis to form the haploid gametes, but the males, they will go under mitosis to form the gametes. Why?
because they don't have to reduce, no?
So they don't have to reduce now because they themselves are haploid, and being haploid, they have to give rise to haploid cells. If they undergo meiosis, so this thing, the chromosome number of the gamete will become n by 2, half of n.
Yeah.
So we don't want this, right? The gametes have to be haploid. So that is why they undergo mitosis. What was so difficult? You were like there. You like, I'm just near the finishing line, but I will not go there. Understood this whole entire concept. So now I will believe that your haploid, diploid, meiosis, mitosis concept is crystal clear. And now from now on, in any other chapter, if I ask any type of question related to mitosis and meiosis, and if you people do wrong or give me wrong answer, then you will see. Up until now, any doubts? Okay. Marwa, do you have any doubts?
No.
Okay. Now moving on to the next topic, that is mutation. Now mutation, it is a sudden change, okay, in the chromosome and the genes, and it is discontinuous and it is heritable. This can pass on from one generation to another. So it is a sudden discontinuous variation in the genotype and the phenotype of the organism. Genotype means in the genes level, phenotype means even in the morphological level. So because of the changes in the chromosome, the gene, like it suddenly happened and no one knows about it. Okay, so you can also call it as alteration of DNA sequence. So what is mutation? It is when the DNA sequence gets altered. Either something gets added to it or something gets removed from it. In both of the cases, we will call it as diffusion, I'm saying, mutation. Okay. Now, mutations, they are of three types: gene mutation, chromosomal aberration, and genomatic mutation. First, we will talk about the gene. What is what is a gene, students? What is a gene?
Functional unit of DNA.
Very good. Yes, it is. Gene is a functional unit of DNA. Students, don't you see that when you give me the right answer, do you see my smile? So learn from my smile. I become so happy from the inside when you people give me the right answer. It's like I feel like, oh, they are studying. Okay. So when I talk about the gene mutation, gene is the functional unit of DNA. So when any kind of change in that gene is going to happen, maybe something is being added to it, something is being subtracted from it. So we'll call it as a gene mutation. So it is the alteration of DNA due to the change in the nucleotide sequence. Gene mutation may occur due to a change in the single base pair of DNA if only one base pair. Do you people remember adenine, thymine, guanine, cytosine?
Yes,
If
ma'am.
you remember, biomolecules chapter, the
Yeah.
dreaded chapter from class 11. है ना? So in that we saw nitrogenous bases. I will quickly get you up to speed. So nitrogenous bases, they are of two types. What are the two types? Can anyone tell me? If you remember. Jesslyn, you too will remember. Please tell. They start with P. Both of them start with P. No one. Ma'am, we are not in 11th. Why should we remember 11th? Huh. Say.
Something with PUY, so I forgot, ma'am.
Purine and pyrimidine, right? So nitrogenous bases, they are of two types, purines and pyrimidines. Okay. Now, adenine and guanine, these are the purine. Thymine and cytosine, these are the pyrimidine. Always remember, a purine pairs up with pyrimidine. So if I say adenine is going to pair up with thymine, how many bonds are going to be present in between them? There will be two bonds, and these will be hydrogen bonds. Same way, guanine is going to be present with cytosine with 3 hydrogen bonds. Okay. Now, if like normally when I say DNA, I think almost all of us think about this, isn't it? When I say DNA. But if you go to the molecular level to think about DNA, it is in terms of these nitrogenous bases. Like it is repeated n number of times throughout, like a coding sequence. So if I say this adenine is present over here, if I remove it, so now the coding sequence will become changed. This is what mutation is. Or if I add some over here, then again a change is happening. So this is what mutation is. Clear? So here we said that if only one base pair or if only one sequence is getting altered in some way or the other, then that is called as point mutation. If more than that is happening, then we call it as a different kind of mutation. Okay, where did all my handwriting go? So now, point mutation, all of you have understood. It's like 1 point, like change in only 1 point, one particular point. That is what we call as a single base pair mutation or a point mutation. Then comes the frame shift mutation. Now see, what happens if there is this sequence of DNA like this, ATG, CGT, ATTGCT. Okay, like this, anything randomly. Now, in our body, like how a computer runs the code, isn't it? It reads the code. Same way, if here we have to, read the code in our body. So how does it happen? It is always read in the triplet form. Triplet like this. ATG, then CGG, then TAT like that in triplets. So for example, if I remove this part or this part. Now the reading frame will be changed, isn't it? It will be ATG. Now instead of being CGG, it will become CTA. And then instead of being TAT, now it will become TTG. Right? Isn't it? One shifted or two places it has been shifted. So the reading frame is now shifted. So the kind of mutation because of which the reading frame gets shifted is called as frame shift mutation. Now, frame shift mutation can be because of deletion of something or insertion or addition. Now, then comes substitution mutation. Now, substitution you people understand. For example, in your school, if your teacher, in your school or in general, your main teacher is absent, in the place of that teacher, another teacher comes, right? That is substitution. Same way, here mutation can happen because of substitution. Now remember that whenever a purine molecule gets substituted by another purine molecule, we will call it as transition. Or when the pyrimidine will be substituted by a pyrimidine, we will call it as transition. Getting my point? So this is whenever the purine is getting substituted by another purine, or a pyrimidine is getting substituted by another pyrimidine, this process is called as transition. But if a purine is being substituted by a pyrimidine and a pyrimidine is being substituted by a purine, then we call it as transversion. Did you people get this thing? Yes or no? Shall I speak it again? You did not get it? Okay, wait. See, I told you that adenine and guanine, these are purines, right? And I told you thymine and cytosine, these are pyrimidines. Now, if in a sequence, the adenine is present, which is a purine. Now, let's just say instead of adenine, there came along guanine. So, the purine got substituted by another purine, right? Or if I say thymine is present initially, and then instead of thymine, cytosine came. So, pyrimidine got substituted by pyrimidine. So, this type of mutation wherein, like this type of substitution mutation, wherein the purine will be substituted by another purine, the pyrimidine will be substituted by another pyrimidine. This is what we call as transition mutation. Got it? Now, adenine is a purine. Now, if the adenine is getting substituted by thymine, now first it was a purine, then came the pyrimidine. Now you will call it as transversion. Is it clear now?
Yes, ma'am.
So that is how the process of substitution mutation takes place. Now remember that the gene mutations, they are spontaneous. Spontaneous means it can happen, you can say, because of, internal reasons, and anytime, anywhere, at any point. It doesn't inform anyone. It doesn't give a signal. It just happens. Many a times it gets unnoticed. Now, despite of it happening naturally and because of the internal structure or things, the mutation can be caused also. Like we can induce mutation also. How? With the help of mutagens. Now, when we talk about what are mutagens, so these are the mutation causing agents, the agents that cause mutation. Now, this mutagen, they can be of two types, physical mutagen and chemical mutagen. Clear? Now, the physical mutagen, they are mostly radiations. You might have heard, or have you people had X-ray done ever? No, you have. So, like for all of you who might have had X-ray done or might have seen someone's X-ray being done. So, firstly, the person who goes for the X-ray, that person goes, and the person that is for the help, right? The guide or anyone, they wear a vest. They wear a very thick vest and they protect themselves. Why? Because X-ray is a very harmful ray that can cause mutation. So they have to protect themselves. And those people that are undergoing X-ray, they have to be exposed so as to find out the source of their problem or the X-ray, like the bone and everything. But radiations are physical mutagen. These are like the ones that cause mutation. So radiations, they are of two types further, ionizing radiation and non-ionizing radiation. Now, ionizing radiations, what are the examples? X-rays, gamma rays, alpha rays, beta rays, all of these different kinds of rays are ionizing radiations. Whereas when we talk about non-ionizing radiation, so UV rays. You might have seen, students, when you go out in the sun, there are some of us that get so badly tanned, isn't it? Like you step out, you'll have that tanning all over. Like I wear hijab, so like I wear the cap also when I go out. So what happens is that the whole entire cap area, I have a line on my foot because of the tanning. So, tanning is a form of skin mutation. It is a form of skin mutation that is being caused because of the UV rays that comes from the sun. Okay. So imagine that is why people ask, you might have seen ads and everywhere, people telling to wear sunscreen, right? So as to protect yourself from the harmful UV rays, because now we know that the UV rays are mutagens. They are mutagenic, right? So in the long run, it can cause skin cancer also. Okay, so that is why now if you people don't, please wear sunscreen, cover your face, and then you go out. It's very, very hot. Now, this much is clear about the mutation, mutagens, you people. Any doubts anywhere?
No, ma'am.
All right. Now, there is also chemical mutagens. Now, chemical mutagens, they are so much more injurious as compared to physical mutagens. Like physical mutagens, they basically, they take time to cause mutation, and they cause mutation little by little. But if you think about chemical mutagen, one exposure and that's it, mutation is caused. Now the mutation can be big also, small also, but some of the examples of the chemical mutagens would be nitrous acid, acridines, base analog, all of these. In experiments, when you go to higher classes, you will have certain experiments in which certain dye will be used. Those dyes, some of those dyes are also mutagenic. So it's like they ask us to wear gloves to do that experiment. Okay, students. So that is how powerful the chemical mutagens are. Now, after that, there comes the next topic, which is polygenic inheritance. Poly means many. So when many genes are responsible for the inheritance, it is called as polygenic inheritance. For example, we saw when we were discussing about the round yellow plants. So round was like represented by two capital R's, and the yellow was represented by two capital Y's, isn't it? But if you will ever see the inheritance of human skin color, now if you go around the world, and you see different people, all the different races, all the different countries and places, like even in India, you will find so many different varieties of skin color. There are people that are extremely white. There are people that are extremely pitch dark, right? So one person is extremely white. It's like white screen. And then there are people that are extremely dark. And then in between those two extremities, there are so many people of different colors, isn't it? Some people are brownish, and in the brown also you have so many shades of brown, right? So remember that the human skin color is not influenced by one, by two, but three genes. So if capital A, capital B, and capital C genes are present, then that person will have the darkest skin tone. And if small a, small b, small c is present, so that person will have the lightest skin tone. So whenever a phenomena or whenever any trait, it is controlled by three or more than three genes, we call it as polygenic inheritance. Now, one such thing about this polygenic inheritance is that every allele A, B, C and their effect is additive. For example, if a person, now we talked about two extremes. This is one extreme and this is another extreme, right? Darkest, lightest. But if I say this, now this person will have a mixed skin tone, isn't it? Neither will he have a very dark skin tone nor a very light skin tone. It will like in the middle. If a person is like this, then also it will be fair side, a little of brownish, right? Like that. So the kind of inheritance wherein so many genes are actually, the effect of these genes are additive also. And other than that, it is, like inheritance is because of three genes or more than that, we call it as polygenic inheritance. Other than the human skin color, there's also human height that is, done like this, human skin color, and also the kernel color of the wheat. Clear? Now, after that comes pleiotropy. Pleiotropy. See, whenever one gene will be showing multiple phenotypes. For example, if I talked about capital R, capital R was responsible for showing round seed, isn't it? Was it responsible for showing any other seed or any other type? Other than the seed shape, was it responsible for showing any other thing, any other phenotype? No, right? So when one gene will be showing multiple phenotype, we will call it as pleiotropic gene. For example, phenylketonuria. This is a disease. Okay, the name of the disease is phenylketonuria. Now, this disease is caused by the mutation in the gene that is being coded for the enzyme called as phenylalanine hydroxylase. So normally in a normal person, phenylalanine hydroxylase has to be synthesized. But because of mutation, not enough of this particular enzyme is being synthesized. Now, because of the absence or because of this mutation in the enzyme, the pleiotropy effect will be shown by mental retardation, reduction in the hair, and also skin pigmentation. All of these three will be the phenotypic expression because of one mutation. So when one gene are showing several different types of phenotypic expression, we call it as pleiotropy. Got it? Okay, then comes starch synthesis in the case of pea plant. So that is also a, like it is an example of pleiotropy. Clear, students? Now, after this process, all right. After that, now we will talk about two different types of diseases. Okay. First set of diseases that we will be talking about, it's because of mutation only. Mendelian disorders. Why we have given Mendel's name with disorder? Is because he was the one who taught us genes and everything. So now everything that will happen because of gene, we will name him. So Mendelian disorders are those disorders that are caused by the mutation in the single gene. So since the mutation is causing a disease and mutation is happening in the gene, so we will refer to it as Mendelian disorders. What are they? The examples: hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, and thalassemia. Have you people heard any of the diseases names before? Do you know anyone's symptoms? Anything you know about any disease?
Can you repeat your question?
The question is, have you people ever heard about any of these diseases? Do you know the symptoms of any of these diseases?
I know color blindness when you're not able to see the colors like
It's not that they are not able to see the color, it's that they cannot differentiate between the colors. Okay, very good. And you people can also say that, ma'am, phenylketonuria, we just saw. Mental retardation, that happens. Right now, only all of you saw. Other than that, all of these diseases are the examples of Mendelian disorders. And we have to study all of them. Okay, how they are getting inherited. Now see, the main thing. The Mendelian disorders, they are of two types: sex-linked recessive, sex-linked dominant. Meaning, in the dominant case, you people know dominant and recessive, right? Recessive will be expressed in one condition. And what is the name of that condition, students? The condition in which the recessive trait gets expressed is called as what is it called as? No one. Homozygous
What
recessive.
did you ask?
Huh? I have said the answer. See, the question is, in which condition will the recessive trait express itself? So it would be homozygous recessive condition, right? Now let us see. All of these different Mendelian disorders, they have their nature, whether they are expressed in a recessive condition, whether they are expressed in dominant condition, and whether they are linked with the body cells or the body chromosomes, meaning the autosomes, or whether they are linked with the sex chromosomes. Okay, accordingly, they have been classified. Let us see. So the first one, that is hemophilia. So this is a recessive disease and it is an X-linked disease, meaning the X chromosome and its inheritance will be responsible for hemophilia. Okay. Color blindness is also X-linked disease. It is not linked with the body chromosome, rather it is linked with the sex chromosome. Then comes sickle cell anemia. Now, sickle cell anemia, it is autosomal recessive disorder. Autosomal recessive. Okay. Autosomal means it is linked with the body cell and not the sex chromosome. And other than that, it is recessive, that it will be expressed in homozygous condition and not in heterozygous. Phenylketonuria is autosomal recessive. Cystic fibrosis is also autosomal recessive. Thalassemia also is autosomal recessive. But there is a disease called as myotonic dystrophy, and it is autosomal dominant disease. Now let us see the inheritance of
Can you repeat
color.
that? Myotonic dystrophy.
Myotonic dystrophy, this is the only disease that is autosomal in nature but the dominant version, meaning it will be expressed in the dominant condition. Whereas all the other autosomal diseases that we have seen up until now, they are expressed only in the recessive condition, meaning only in homozygous condition and never in the heterozygous condition. Now see, the first one that we will be studying the inheritance of is color blindness. Now, during the color blindness, it is an X-linked recessive disorder, right? So this is an X chromosome and the gene that it will be carrying will be the C. C refers to the color blindness. Okay. Now, those people, okay, wait. What is the nature of color blindness? It is X-linked recessive, right? People understand this term? Now tell me, now, in all of these four situations or all of these four combinations of genes, in which one, or I'll number it 1, 2, 3, and 4. Which one out of 1, 2, 3, and 4 is a colorblind person?
The first one.
The first one. Why?
Because both of them have color blindness, so the offspring also will have color blindness because it's dominant parents.
No, not because of the dominance. It's because right now it is in the homozygous condition, isn't it? In the second case, the person, the female, is not colorblind, but is a carrier for the color blindness gene. Because one of her X chromosome has the C, but not both of them. Third, the male is a carrier, but the fourth male is unaffected. Clear? So now, in this case, C, XCY is affected male, right? XC is affected female, and XCX is a carrier female. Now, can someone tell me why is XCY an affected male? Why is he not a carrier? Why is he affected?
You might understand.
See here. Here, the male that carries the color blindness gene in the X chromosome, that male is said to be affected. If you look at the third situation, the female that carries one of the gene is a carrier. So why is male affected and female a carrier? Come on, you can do it. You can do it. You can tell me the right answer. Which type of disease is color blindness?
It's genetics.
No, no. What I have told you. Tell me that. Yes, it is a genetic disease. That's true. What is the nature of the disease? It is an X-linked recessive disorder, right? So why is the male affected and the female is a carrier? Why this discrimination? I gave you the biggest hint in the world. I couldn't have given a bigger hint than this.
Mom, because maybe the DNA, there's a mutation in the DNA.
This is happening because of mutation only. I'm not asking that. I'm asking in terms of the genotype. You tell me. It is an X-linked recessive disorder.
Because X is the defected gene.
X is defected, but it's defected in the female also. So why is the female carrier but the male is affected? It's so simple that when I tell you,
Ma'am, because maybe because women have XX chromosomes.
yes, exactly, because the females, they have two X chromosomes, right? But the male doesn't have an extra X chromosome. The male has one X and one Y. That is why, since it is an X-linked disease, X-linked meaning it is X chromosome linked, and the male is going to have X chromosome, one X throughout its life, throughout his life. That is why the male having one X chromosome that carries the color blindness gene is affected. Whereas the female, when she has one, it's not affected because she has another X that is healthy. Got it? Did everyone get it? It is an X-linked disease. That is why. So, in this case, what happens is that the inheritance of color blindness is called as criss-cross inheritance. How? Because here, a male is going to transmit his trait of color blindness to his grandson through the daughter, and the female is going to transmit it to the granddaughter through her son. It's like crisscross inheritance. Now, color blindness is a sex-linked recessive trait in which the affected person fails to distinguish or differentiate between the colors red and green. They can't clearly make out which one is red, which one is green. Clear?
Yes.
Now the cross. Let us see what are the possible reasons or the ways it could happen. So when a normal woman that has XX chromosome genotype marries a colorblind man, so now their offspring will be like this. Two of them will be the carrier females and two of them will be the normal males. Like if you do the crossing, you will find this out. So whenever a normal woman is going to marry a colorblind man, these will be the possible outcomes for their offspring. But if a carrier woman is marrying a colorblind man, now do the cross, students. Tell me, whenever a carrier woman, carrier means she will be having this and the male is colorblind. So like this, right? So what will be the possible offsprings? An affected female, affected male, carrier female and unaffected male, right? These will be the offspring, right? So like they can give you to make out the different this thing inheritance pattern. They're like a carrier woman, a carrier for color blindness marries a colorblind man. What will their offsprings be like? They can ask you. So you have to do this crossing and give the answer. Clear? Any doubts up until now, students?
No, ma'am.
Okay. Now tell me, what is the last thing you people note down from this chapter? What is the last thing you people note down? Just tell me quickly. Monohybrid cross was completed, right?
No, ma'am. We did the limitations, I think, of the. Yeah, we did that.
Limitations of?
Mendel's law.
Oh, still there. Like, why was Mendel's law not recognized?
Yeah.
This blood group is done.
No, no, no.
This is not done. So limitation part is like later on. These different headings are done. Terms.
No, no, no.
No. What is the last thing you people tell me? I'll scroll till there only.
Ma'am, from 6 number before genotype.
You have to
Yes.
start. You have to start from genotype.
Yeah.
Okay. Everyone has to start from there only, right?
Yes, ma'am.
You people take down the notes now. When I ask you what is the last thing you people have written, you go blank. Is it the case that you don't take down the notes? You do, na? Okay. Okay. Now you can note it down.
Can you scroll down? Done, ma'am.
All of you are done, students?
Yes, ma'am.
Done, ma'am.
All of you are done, students?
Yes, ma'am.
Okay, Marva, are you also done?
Yes, ma'am.
Okay.
I'm done.
Then students,
Yes,
Yes,
ma'am.
ma'am.
Done, students? Shall I scroll?
Yes.
Okay, students, let's just keep it till here only for today's class. And I will send you the complete notes. Okay. And we will hopefully complete this chapter by the next class. Okay.
Yes,
Till
ma'am.
then, wherever you people have any doubts, do let me know and we will discuss it. Okay. Then we will solve the questions related to this chapter because that's very important. All right, students.
Yes, ma'am. Thank you, ma'am.